This is just a little supplement to our notes on transcription in protein synthesis. So I wanted to show you this picture again because this is important. Make sure you know the difference between prokaryotic and eukaryotic protein synthesis. Now, one of the major things that you want to notice here, obviously, is that eukaryotic cells have a nucleus. So transcription and translation are going to happen in two completely different steps, in two completely different areas of the cell. In prokaryotes, they actually can happen at the same time. You can literally have an mRNA coming off of the DNA and feeding through a ribosome and building a polypeptide protein. So transcription and translation can be almost like simultaneous in a prokaryote. In a eukaryote, they're obviously very, very separated because the mRNA has to leave the nucleus to get to the ribosomes for translation. The other thing is this processing step. In mRNA, you make a pre-mRNA, and then it's going to get a cap and a tail, and it's going to have introns removed, and the exons are going to be spliced together, hooked together, and then that is going to leave the nucleus and go on to translation. So I wanted to clarify that, make sure you know the difference between the two, and that's actually going to come up when we get to applied genetics or genetic engineering. You can't just take a gene from a eukaryote and stick it in a prokaryote and expect it to work, because eukaryotic genes have introns and prokaryotic cells don't, so they wouldn't know how to read that DNA, so we're going to have to include some extra steps. Okay. Again, I already have notes on this, but I wanted to clarify this particular slide. So there's these transcription factors we're going to learn about in another chapter a little more, like switches that tell the DNA, hey, we need to activate now, we need to make this particular protein. Because remember, even though you have 46 chromosomes in every cell, not every gene on every chromosome is making protein all the time. You're making only proteins when you need them. So genes are going to get activated and deactivated as needed. So these transcription factors are going to set up the scenario so that RNA polymerase can come in, and it's going to copy a section of the DNA, and it's going to make an RNA, an mRNA match to that one side of the DNA. Now, this was not really clear in my original lecture, so this is what I wanted to show you. So imagine this is my DNA, and my DNA opens up in this one little section, and it's going to make an mRNA copy. Now, the side of the DNA that's going to get copied is going to be called the antisense or the template strand. So this is my template that my RNA, my mRNA, is being built from. The other side of the DNA, the side that is not being copied, is sometimes called the sense strand or the coding strand, which sounds really confusing because it's like, well, if that's the coding strand, why aren't we reading that side? So let me show you why. And hopefully this will help it make more sense. So imagine I have T, C, C, G, G, let's say A, okay? My mRNA is now going to get built from this. So now my mRNA is going to be built using this template of DNA, right? So this is going to be A, G, G, C, U. Why U? Because there's no T in RNA, remember. All right, so I have AGGCU as my RNA. Now, here's the reason why this side is called the coding strand. What's going to be the code on this matching side of DNA? Well, T would go to A, C to G, C to G, G to C, and my last one here would be a T. Isn't it true that this side of the DNA that was not being copied by mRNA is actually the same code on the mRNA we just made? AGGCU, now it does have a U instead of a T, AGGCT, same. So this is sometimes called the coding strand because technically this is going to be identical with the exception of having T instead of U. This is going to be identical to the mRNA that's made. So this is called the coding strand. You could actually look at this side and say, oh, well, my mRNA that's going to be made from this side is going to be AGGC, this would be a U. So I don't know if that helps, but that's the reason why it's called that. There's also a promoter that tells it where to start and a terminator that tells it where to stop. And there's an area called the TATA box that it contains this promoter. Again, this all goes along with the transcription factors, the idea that we're not copying the entirety of DNA, both sides, top to bottom. We're copying one gene on one side of the DNA. We're making just a template of that in mRNA that's going to now go to the ribosomes. All right, and this diagram shows the same thing. Template or antisense strand, this is the side the mRNA is copying, and the code that's on this mRNA will actually be identical, with the exception of U instead of T, to the sense strand or coding side of the DNA. Okay, in prokaryotes, again, there's no modifications. That's the end. Transcription happens, it's over, and translation can begin. In fact, it may even begin before transcription is over. But in eukaryotes, there are going to be modifications. And again, I believe this is all in my original lecture, but I was just going to comment on this part one more time. So it's going to get what's called a 5' cap. It's just imagine this is my mRNA. It gets basically a cap at one end and a tail at the other. These help it to leave the nucleus and not get broken down. mRNA is temporary. It's not going to stay forever. You're going to make it. It's like having a message that's going to self-destruct in 20 minutes. So the message goes out, you read the message, the message self-destructs, and then you could build a new message. The RNA doesn't stay forever, but this cap and tail will help it to leave the nucleus and not be degraded immediately. And then it's going to get spliced or cut and sealed together. Splice actually would be to seal two things together, not really to cut. So the spliceosomes, they're going to come in and they're going to cut out pieces of the RNA that are not going to be part of that particular protein. Now, important, it can be what's called alternatively spliced. Imagine you have a sentence, and I take some words out of the sentence. I could actually change the meaning of the sentence, right? If you take the word not out of the sentence, you would completely change its meaning. So what can happen is you can have a gene, two genes that sort of overlap each other, and they code for making the same original mRNA, mRNA1, mRNA2, and this is coded for by gene 1, gene 2. But what can happen is this one, gene 1, this mRNA, this section gets cut out, and this section gets cut out, and this section gets cut out. But gene 2, the mRNA that's made, maybe it cuts this section and this section. And these introns just get recycled, they get cut out, and then exons will be spliced together with ligase. Put these together, and this will be my final mRNA, and it'll be shorter than my original. So if this is my original DNA, it gets copied to an original mRNA. The introns will get removed. This will also get, remember, a cap and a tail, and the exons get sealed together, and this will be the final mRNA that's going to go to the ribosomes and produce a protein. So I just wanted to clarify that. This shows, again, the cap and the tail. This step is called processing, and that's going to happen before the mRNA leaves the nucleus. So the final mRNA may actually be significantly shorter than the primary mRNA that was originally made.